All right, well, welcome everyone to the NIAMRI December Member Spotlight. Before we jump into the presentation today, just a handful of reminders. Your audio line is muted. We'll take questions at the end of the presentation, and if you have a question that comes up during the presentation, please use the Q&A option at the bottom of your screen to type in questions there. This webinar will be recorded and we'll be posting it on NIAMRI Exchange later on this week. And so it's my pleasure to introduce our speaker today, Jim Miller, who's the Chief Executive Officer of Atterx Biotherapeutics. Atterx is a specialty pharmaceutical company focused on developing products for the prevention and treatment of multi-drug resistant infections. So we're very happy to have them as part of NIAMRI. Jim's got a background in investment banking and capital raising roles, as well as an MBA in finance. And he's been involved in a number of startup companies. We've chatted a little bit over the time, and he's really done some great work with other startups, and comes into the startups with a role in, a background in economics from the University of Virginia. So Jim, I'm going to stop sharing my screen and turn it over to you for you to go ahead and start the presentation.
All right. Well, thank you very much. Thank you for the introduction. And thank you for the opportunity to spend some time with you today to introduce you to Atterx and what we're up to. I'm going to spend a little time talking in particular about the products that we have in development and some of the history of those products, particularly some of the studies that have been done with these particular products in both humans and animals across two of them. But Atterx, what we're all about is developing clinical innovations for the prevention and treatment of multi-drug resistant bacterial infections. As said, I do like starting out my presentation with this quote. I am giving a lot of presentations to potential investors, and I'm constantly surprised how few people really understand how big this problem is. And obviously this audience is very well aware of it, but this particular quote where it says the problem is so serious that it threatens the achievements of modern medicine in a post-antibiotic era in which common infections and minor injuries can kill is a very real possibility. This is kind of a wake-up call to some of my audiences when I give this presentation. So I did go ahead and keep it in this presentation, even though, like I said, the audience is well aware of the problem. But not everyone is. And so it can be a real eye-opener to some. So as was already shared, Atterx is a private development stage specialty pharmaceutical company focusing on proprietary products for the prevention and treatment of multi-drug resistant infections. The Atterx technology platforms that we use focus really on two technologies, one utilizing bacterial interference and another one using conjugation-killing. We do have two products right now that are farther along in development. As I'll share with you in a minute, we actually have three, but the two that we're really focused on right now, our lead product that we call C1205 is a product that uses bacterial interference for the prevention of catheter-associated urinary tract infections, a big problem in hospitals, which we'll chat about a little bit more later. But bacterial interference is very much of a powerful approach for preventing UTIs, and it does minimize antibiotic use. And with the approach, an innocuous bacteria are allowed to colonize in the bladder and thereby competitively inhibiting the growth of the UTI causing pathogenic bacteria. That's our first product, C1205. Our second product, GN4474, is a topical that's used for really the treatment more than the prevention of infections and burns, trauma wounds, other type of wounds, low perfusion wounds, and the rest. And this product uses conjugation-based killing technology to target the burn and skin infections that are caused, but in this case, by gram-negative bacterial pathogens. So 4474 focuses on the treatment of the gram-negative bacterial infections. To give you a little idea of our pipeline, so a little bit of a visual graphic here, C1205 is actually getting ready to begin its phase 1B portion of its combined phase 1B, phase 2 clinical trials. So we are beginning the enrollment as we speak in site selection. So that product is really now into clinical trials. Our second product that I mentioned, 4474, is a little farther behind. Right now, it is actually more in the determination of safety and efficacy, primarily in animals. I will share a little bit of some data from some animal studies that were done in mice with this product, but we also right now are gearing up to do a large animal study with two different academic institutions. And that's where we are with that product, getting ready to get a little more data, as I'll share in a minute, on some of the larger animals. In this case, it'll be a pig study. And then we do actually have a third product out there. It is another topical using conjugation-based killing technology, but this product will address and treat gram-positive infections. So it's a bit farther behind. This one's still, what we like to call, is still on the bench, but we are getting some real interest by some third parties to do some co-development on this product as well. So just an overview of the two products, a little bit more detail on them.
Jim,
Our C12,
sorry to interrupt. It doesn't appear that
Sure.
your slides are advancing.
They're not. That's strange. They're not. Should we go back to working with you?
Sure, if you want, I can go ahead and share from my side.
It's they're advancing on my side and it shows that I'm sharing, but why don't we stop?
Okay.
And if you want to bring it up, I'll do a little bit of a rewind. So did it just stay on the first slide?
There we go. And we saw this slide, the pipeline slide.
Okay.
So would you like me to advance one more?
Yeah, let's advance. Okay, here is the just a little bit of an overview, the technology overview of our two products, C1205 for the prevention of urinary tract infections. As I mentioned, this is simply introducing a harmless bacteria. It happens to be an E. coli that effectively prevents the infections from the pathogenic bacteria. In simple terms, it basically outcompetes the pathogenic bacteria for the limited nutrients in the urine once it colonizes the bladder. It's very effective. We actually have data from human subjects, safety and efficacy data on over 220 patients, both in Europe and the US. But we do know through these studies that it does prevent the catheter-associated urinary tract infections. And then 4474, this product's a bit different. It also utilizes a harmless bacteria, but this delivers a killer plasmid into the pathogenic bacteria using the natural process of conjugation. And we have found that 4474 is highly effective against all gram-negative bacteria, including the escape pathogens. The unique part about this product is that we have been unable to get the several of the pathogens to become resistant to the product, which will show a little bit of study results on that as well. So next slide. So again, we're going to go ahead and start with C1205. Just a little bit of data here on catheter-associated UTIs. Many people don't know how big of a problem this really is in the hospitals, and there are 1.7 million hospital-acquired infections annually in hospitals, and 36% of those hospital-acquired infections are urinary tract infections. It's the largest percent of hospital-acquired infections are urinary tract infections, but 80% of those are due to catheter use. And what a lot of people don't know is people die from UTIs or complications of UTIs, and we believe this number is way underestimating, but they do say up to 45,000 deaths due to complications from UTIs each year. One of the key things from an economic standpoint and a business model standpoint is CMS no longer reimburses hospitals for the treatment of catheter-associated UTIs. And so for our standpoint, from a business approach, the prevention of a catheter-associated UTIs can save a hospital a lot of money, and they will get reimbursed for the use of our product for the prevention, where they are not being reimbursed when they, in fact, have to treat a catheter-associated UTI once it's started. Next slide. So here's what our product looks like. It's a lyophilized, freeze-dried biologic. It's resuspended with water when needed at the bedside to form a therapeutic lubricating gel that's the same consistency of KY jelly. It's applied to the urinary catheter just like the KY jelly would be prior to insertion in the patient. Next slide. So just a little bit more of a graphic, and then we also have an animation I'm going to share here in a minute. But again, what's happening here is this harmless E. coli is being introduced into the bladder via the catheter in the gel. It quickly colonizes the bladder and again, outcompetes the pathogenic bacteria for the limited nutrients in the urine. It is a very stable lyophilized biologic that has a very long shelf life. We have established human safety and efficacy. And the key thing here that I wanted to share is we recently have had our IND approved. And so we are ready to begin our combined phase 1, phase 2 clinical trials with this product. So I believe the next slide is the animation. So they say a video is worth 1,000 words. And so here is just a video representation of our product. Our product is on the gel, on the catheter. Once inserted, it quickly colonizes the bladder. And in doing so, it outcompetes, again, a little repetitive here, but it outcompetes this pathogenic bacteria from competing and taking hold because the colonization outcompetes the pathogenic bacteria for those nutrients. So if there is a pathogenic bacteria that might have been introduced through the insertion of the catheter, they quickly die off. And that's one of the things we are going to be showing through our clinical studies is how quickly the bladder does colonize, how long it stays colonized, and critical to that. So next slide. Oh, I think you have to double click. So here I wanted to talk a little bit about previous bacteria interference clinical studies that have been done that have involved two closely related strains of benign E. coli. The first on the left is E. coli 83972. This is the wild strain of the E. coli, and this was actually isolated from a young woman in Europe quite a few years ago. And she was just asymptomatic, had the bacteria in her bladder, but was not showing any signs of infection at all. And so this has been studied pretty extensively. These studies done in Europe, but it really did establish the safety and effectiveness of it. Our product in our E. coli, we call it our E. coli, is actually it's E. coli HU2117, and it's actually identical to 83972, except there has been one gene deletion. There's been a gene deletion of a PAPG, so that there will be no pili, form, which will keep there from being immune response. Now, what we did is we felt just to be on the safe side to remove any pili or the formation of any pili, because that's what causes the adherence to the bladder and can ultimately cause some issues. But it turns out that even the wild strain doesn't develop pili. So it is an extra step caution, but our product is actually a formulation of E. coli HU2117 that includes in its formulation the gel and lubricant components and that type of thing. But in this case, there were studies done at the Baylor College of Medicine on the effectiveness of the reduction in catheter-associated UTIs, which I'll share those results in just a minute. But again, because of these results on these studies that were done, the FDA did two things for us. The first thing they did is they agreed to combine the phase 1 and phase 2 into one combined phase 1B, phase 2, which really does reduce the time to reach the end of a phase 2 study and reduces the cost as well. But the other thing they did is they agreed to, instead of upwards of 200, 250 patients to be part of this study, and because we had all this data on the human patients through primarily the Baylor College of Medicine, they agreed to a much smaller cohort. And so as you see down in the darker block, Atterix did have our IND reviewed and approved by the FDA, but the phase 1 trial is using our formulation. In this case, it's really to determine some of the colonization, the rate of colonization, along with safety and effectiveness. But that's just 10 subjects is all that's required. And then we're going to be able to move very rapidly into the phase 2 section of the study. And that's going to be with 30 patients that we'll be following for about 6 months as well. And the main focus, as we mentioned, is to evaluate all aspects of the bladder colonization with E. coli HU2117, rate of colonization, length of colonization. Those things are really critical to the assumption that it in fact is preventing a UTI from forming. Next slide. So I want to focus on the studies that were done at Baylor. So at the Baylor College of Medicine, there are 4 main studies that were done, and 3 of them demonstrated very quickly that the bacterial interference strategy was working, but we were having a little bit of difficulty with establishing some of the metrics that we wanted on colonization because it was using bladder installation alone. So the installation technique was actually being used. So they were taking freshly cultured and washed bacteria, suspending in a saline, and then inserting into bladder. And so that insertion process was a bit not only barbaric, but it was causing difficult results on whether or not colonization was happening to the level that we wanted. So 3 studies were done around that still showed that it reduced the incidence of catheter-associated UTIs in the study subjects. But the last one, the 4th one, is the key publication that really got us moving forward on the development of our own product, C1205, figuring out how to make it a shelf-stable product. And that's why we chose the lyophilization with the reconstitution with water at the bedside. But this one actually is a study where a gel was developed and placed on a catheter, and the catheter, of course, inserted, and the study subjects monitored with that. So in the next slide, to show a little bit of the results, on the left-hand side, this is the wild strain. So even with the wild strain and some of the challenges with the bladder installation technique, if you will, we still had dramatic drop in the incidence of the catheter UTIs on a per patient year with the subjects. So the 2 for E. coli 83972, and the first one on the left with the E. coli HU2117, again, those were utilizing the installation processes where the study 11717, I have to double check with my reading glasses here. It's a little small even for me. So in this case, though, when you look and you see the number of UTIs per year of 9.9, that's almost a UTI a month. And these were, the unique part of this study is this was all spinal cord injury patients with neurogenic bladders. And so you saw that with those patients, there is just a higher incidence of chronic UTIs due to catheter, due to indwelling catheters. But in our study, we were able to reduce that to less than 1 a year. So very dramatic. So once we were actually able to begin focusing on neurogenic bladders, also working with spinal cord injury patients, and using a delivery mechanism of a gel on a catheter, we started seeing really dramatic effects in the colonization and the cause, and then therefore the significant reduction in the incidence of the catheter-associated UTIs. So next slide. So just kind of wrapping up here on C1205, as we mentioned, we have previous clinical data using the E. coli strain, both the wild strain as well as the slightly modified strain. We do have extensive safety data on the colonized human patients. The bacteria was well tolerated in the patients, and it's been very effective at reducing catheter-associated UTIs. As I mentioned, the FDA approved, it was actually amended IND, and so we are ready to start enrollment phase of the combined phase 1b/2 clinical trials. When I mentioned amended, we did decide after having an approved IND, after working with our FDA consultants, to go back and request a few amendments to the protocols, which allowed us to shorten the time to complete the clinical trials and decrease the cost substantially. And those really had to do with some of the enrollment eligibility to move to patients with neurogenic, all patients with neurogenic bladders, not just spinal cord injury patients, and being able to accelerate the enrollment in the phase 2 component as soon as we've finished the enrollment of the first 10 in the phase 1. So the approved amendments really now have shortened the time and reduced the cost of our combined phase 1, phase 2 clinical trial. The other thing we're doing that I just wanted to share is really twofold, just interesting from a standpoint of a company when we're moving into the clinical trials and the clinical development side of things, is we're actually using a hybrid approach to the management of the combined phase 1, phase 2 clinical trials in order to not only lower the cost, but to have more control. What we found in trying to work with full service CROs is they had a little difficulty being able to adjust their pricing structure to a study that only had 40 patients versus their typical studies that have 200, 250, 300. So working with our FDA consultants, we have designed a hybrid approach where we're actually more involved in the site selection and the training of the PIs, all of that. And the other thing that wanted to just kind of share with the group is we've been working with the National Institute of Health, the National Institute of Allergy and Infectious Disease under a preclinical services agreement. So as we're gearing up to begin our clinical trials with C1205, we need that product manufactured for those clinical trials. And we're working with the NIAID for them to actually do the manufacturing for us in the service in-kind services approach, where basically they'll be doing it for us for free. So across the board, the belief in what we're doing from government, from some of the other things I'll share with you and some of the academic institutions, we're just really well positioned to get moving into our phase 1b of our combined study. So next slide. So now I want to take a few minutes and get into our topical GN4474, again, using conjugation-killing technology. And this one is for the treatment of all gram-negative bacteria. So next slide. So this we found to be effective against all antibiotic-resistant gram-negative bacteria. It is a topical applied to the skin and skin surfaces. Just a couple statistics, as I kind of shared earlier, there's over 5 million patients with gram-negative skin infections annually in the US. Again, we believe that's an underestimation of what really is out there. Over 10,000 people die annually. Again, we think that that is probably quite a bit higher, particularly people, many times the cause of death is not necessarily attributed to the infection itself, it's something else. But as we know, even more recently, that some of these gram-negative or just these antibiotic-resistant infections that people are getting from a simple cut on their leg that's resulting in an amputation or sepsis is causing a big problem out there. And we do like to put the logos of some of the government entities because this product has really been supported by some non-dilutive funding from the government. We've got a lot of interest here, again, from the Department of Defense primarily and the Warfighter program, and really getting the product through the research phase and into where it's now a product ready to go, ready to move into clinical trials. Next slide. So how does this work? I do have a video, but I do like this slide as well. What it is it's a conjugation mediated killing of all gram-negative bacteria. Basically, what we've done is we've engineered a donor bacteria to deliver to the pathogenic bacteria a killer plasmid, a highly engineered killer plasmid with toxins in it. And this is kind of where our IP comes in and how this is designed and effective. But the conjugation occurs between our donor bacteria and the pathogen, and the killer plasmid is simply transferred into the killer plasmid by the process of conjugation. Once in the killer plasmid is in the pathogenic bacteria, because it doesn't have the appropriate repressors to keep it from expressing, the toxin genes are expressed and it kills the pathogenic bacteria immediately. Pretty elegant on how this works, and but it is highly, highly effective. Next slide. So just a simple animation. You know, when we have a wound that is infected here, you know, many times it needs to be treated before skin grafts or other surgical procedures. So our product is placed over the wound, either through just the gel or through a dressing. And once it is there, this is when the conjugation process begins. So moving down to the level here, our bacteria is our donor is the blue, and the pathogenic bacteria is the red. So through conjugation, that killer plasmid is shared with the pathogenic bacteria, and those toxins are quickly expressed, and the pathogenic bacteria is killed almost immediately. It is very, very quick, very effective once it's applied, and throughout the wound. So quickly, the wound is cleared of the pathogenic bacteria, and whether it's a skin graft or surgery or whatever, or even just heading home because you're now have got a clean wound without an infection, all the above. So that's again how that product effectively works. The way that I kind of explain it, because I think we said at the beginning, I do have on my team some, you know, very bright scientists, PhDs, microbiologists, and the rest. And they explain it probably a little bit more scientifically than I do. But for me, it's pretty clear that I use the Trojan horse analogy, that we basically we're faking out the pathogenic bacteria to accept the through conjugation our killer plasmid, and it's expressed and kills the pathogenic bacteria. What we've done is we've tested this against all of the major gram-negative bacteria, the ones that are, you know, the most pathogenic out there, the Acinetobacter baumannii. Once again, seem to always catch up on that one. And Pseudomonas aeruginosa, I've got that one down. But as you can see, the clinical isolates that were used were pan-resistant to most commonly used antibiotics, and all clinical isolates were killed in a single 2-hour treatment with our GN4474. So here, the number of isolates that were tested, all of them were killed through the application of our 4474. In the next slide, oh, go ahead. Here's where we did some studies on mice. So in this case, a group of 26 mice with both Pseudomonas and Acinetobacter baumannii. We burned them, infected them, and on the left, in the case of the untreated mice, all of them died within 2 days. With the right dosage, all of them lived. And then we had similar results, but not quite to the same dramatic degree on the right-hand side. But for me, when I saw this from a standpoint of getting involved with the company, you know, I many times see products that do something a little better than the standard of care. But when I saw something that untreated all die, treated at the right dose all live. That got my attention. And this has been very influential in talking to investor community and the rest when they see something as effective as this, even if it's in mice. Next slide. So we also did try to force resistance with these two pathogens. And if you're familiar with the typical study protocol for trying to establish resistance, these were exposed and repeatedly up to 20 times to try to force the development of resistance. And my understanding here through my scientific group is if it began to develop resistance, these two lines would actually cross, would close in and cross. And the fact that they stay pretty much straight like railroad tracks shows that at least through this number of applications, we were unable to force resistance to our product. So again, this is a big differentiating factor for this particular product and has garnered a lot of interest of several different groups out there, including some academic partners that we'll talk about in a minute. So that is our gram-negative, that is our topical GN4474. So our next slide. So I don't know if we might have skipped our summary for C1205. I apologize if we did. I don't think we did actually, though. What we did find is GN4474 is effective against all antibiotic-resistant gram-negative bacteria. Atterx did delete and added a few genes, but the main thing was the deletion of the PAPG gene for additional safety so that there would be no risk to humans, animals, or the environment. We have not been able to force resistance in our tests that we've done. One of the things I did want to speak to here a little bit more is, as I mentioned earlier, we are moving and are at the stage with this product of animal studies. And we do have the data that I just shared with you on mice, but we believe before we submit an IND to the FDA, we really need data on large animal. So we're actually currently submitting for some SBIR funding, and we're working closely with both the University of Michigan, Michigan State University. Gosh, I could be shot for that in this state of Michigan. Michigan State University Innovation Center, as well as the University of Wisconsin Center for Biomedical Swine Research. In both cases, we're working with them on the development of the study protocol, study design, so that we can submit the SBIR grants here in the next month or two, and are hoping to get some non-dilutive funding to support those efforts. And one of the things Chris had mentioned at the beginning is, how can some of the people who are attending help us? Well, we're still looking for partners on the academic side to potentially work with us on some of our studies, particularly our large animal studies. So I did want to just put a little plug out for that. But that's what I planned on sharing today. I did mention that it usually keeps to about 30 minutes, but I'm happy to answer any questions that anyone might have.
All right, great. Thanks, Jim. Before we start, and there are quite a few questions that have been coming into the Q&A, so just a couple quick announcements to wrap things up here before we start the question and answer. Our next member spotlight will actually be QIAGEN on January 7th, I believe, 2025. So that'll be our first member spotlight for the new year. And then we'd like to remind everybody that registration for the NIAMRI annual conference that will be held May 13th through the 15th next year in Raleigh, North Carolina is open. We have super early bird pricing available now through the end of the year, and the QR code at this screen on the screen will take you to the registration link. So thanks everybody for joining. I'm going to stop sharing my screen and we'll start answering some questions. So the first question has come in, and this is from Nancy Hansen at Creighton. Are you Inhibiting mannose receptors, have you tested multi-drug resistant E. coli? How about ST131 E. coli? And have you tested patients that have recurrent UTIs in the community?
Well, at this
And
point in time, the answer to most of those questions is no or not yet. Our primary focus right now is on indwelling catheters. So focused on spinal cord injury patients that have an indwelling catheter in for usually over 30 days and switching out every 30 days. We are hoping to, down the road, move to more intermittent catheter and therefore more of the recurrent UTI. And we actually know of a few other delivery mechanisms out there that are being done at a couple of different academic institutions that we hope to enter some discussions with and might have some licensing on some of those opportunities. But our real focus so far has really been on the prevention of the catheter-associated UTIs with indwelling catheters.
great. Another question, for a lot of antimicrobial resistant organisms such as E. coli and Klebsiella, they carry multiple plasmids. And since they can carry multiple plasmids, have you seen any host restrictions due to plasmid interference?
No, we haven't. At this point in time, if you can give me the name of the person who asked that question, I'm happy to get Steve Watt, my chief scientist, to make sure that I'm answering correctly. But from my understanding, no, we have not experienced that.
That sounds great. Yeah, I can share that name with you
Yeah.
after the
Yeah,
presentation.
some of the more scientific questions are wonderful, but I've got much higher qualified people on my team to take care of those answers for you.
And we still have a couple more questions coming in. Do you foresee any contraindications for using C1205 in certain types of patients, such as those who might be immunocompromised?
Well, the spinal cord injury patients that we were using, at Baylor, many of them, were compromised, and we were still able to show good results on the reduction of the UTIs, from the 9.9 down to less than 1. But again, that question I think I'd rather pose over to my chief scientist, because I don't believe there has been, but again, that's one where a backup from my scientific side of things would be good. So is there a way that I can get some of these questions and the people who ask so I'd like to quickly answer them back? Because again, as we started at the beginning, my role has been on the fundraising side and doing the presentations. And when Steve Watt, my chief scientist, is the one who really dives in on the science side of things.
Yeah, so that last question came in from an anonymous attendee, but Jim, what we might have you do is maybe drop your email address in the chat, and so folks that are listening could potentially reach out directly to you, and if they have follow-up questions or if you want to connect them with the scientific staff at Atterx, that might be an easy way to do that. I also know that you're in Nyamri Exchange, and that would be another way for our listeners to reach out directly to you.
Yes,
So,
please
Jim,
do.
yeah.
Please do. Again, I've learned to not necessarily speculate and know enough on some of these deeper dive scientific questions and know the general. But to me, I'd rather it be a more productive dialogue between people who are really understanding exactly what's being asked and what's exactly being answered.
Great. So I don't see any more questions in the Q&A right now. We'd invite people to go ahead if you've got any remaining questions, get them entered there. But one that came to mind for me, Jim, is so it looks like you're getting ready for those phase 1B, phase 2 trials. Are you seeking patients for that? Do you have your sites lined up?
No, we are still early in the process with that. We're actually in discussions right now because one of the main eligibility requirements is a patient with a neurogenic bladder, and of course, primarily spinal cord injury patients. We have been talking to some of the top spinal cord injury rehab hospitals, and currently we're talking to Shepherd, we're talking to Research Institute in Chicago, we're talking to University of Miami, but we are still in the process. So if anyone has any ideas of where it might, we might be able to conduct some of the clinical trials, we are very much all ears for that.
Great. And then a question about you. You mentioned, wanting to do some large animal models there with the topical product. So swine looked to be one. Is that the only large animal model you're considering right now, or would there be others?
Right now, we've gone through the process of with particularly working with the University of Wisconsin Research Center there. And it just seems, for a lot of the reasons, including, as we know, pigs have skin similar to human skin, that a pig makes sense. We've talked about actually doing some studies more towards low perfusion wounds and using, in this case, more back maybe to mice and or rabbits. But we have not really gotten far enough along on that, and they're still waiting for some proposals back. In that case, we're looking to the Michigan State University Innovation Center on that. But I do want to just these conversations are early. I'm new to this organization, and it just seems like every time I have some time to look, there's just all sorts of wonderful resources. So again, if there's anything that anyone has any ideas, we are, nothing is set yet either on the site for the phase 1 clinical trial, nor have we fully written any contracts with any academic institutions on the animal studies.
Great. So I don't see any more questions coming into the chat. Very, very interesting presentation today. Really, really elegant approaches to treating AMR and infections. So appreciate you coming in and giving this presentation, and we wish you all the best as you start these trials and continue developing these important compounds.
Well, thank you very much. I appreciate the time everyone took. Now, I am curious. I was looking at some of the questions, and those are questions that we can turn around the answers fairly quickly. I didn't know if there's a way that I can actually copy those and get them back, or if anyone is just can reach out relatively quickly, I can get those answers back in very short order.
Yeah, Jim, I can. I'll do an e-introduction with you and Nancy, and get you connected directly after we're done here.
That'd be great. That would be fantastic.
And these will be here may come through in the recording too. So we would have either the text log of that or on the recording.
Great. And again, as I guess a relatively new member and everything else, I look forward to further interaction with other members and hopefully attend some of the events that sound like they're coming up.
Great. Well, thanks everyone again for joining today. Jim, great to talk with you. Great presentation. Really appreciate your time today.
Thank you so much. Appreciate it. Everyone have a great day.
Thanks, everybody. Bye-bye.